Okay, so in this video, we are going to discuss chapter 7, part B. We are going to continue to discuss on question 5. Okay, question 5. So, question A, the human bodies have different lines against pathogen. So this includes natural barriers that prevents the entry of pathogen to the human body. Describe how tears act as a barrier. Okay, so in this question, it says includes natural barriers. So this it refers to the first line defense, the external barrier. Okay. So for this question, it asks you to explain on how tears act as a barrier. Okay, so the tears will contain lysozyme. Okay, lysozyme, and this lysozyme will destroy the bacterial cell wall. So if you refer to your notes, it is in this slide, okay, which is secretion from skin and mucous membrane. So it includes sweats, mucus, salivas, and tears that is hostile to many microbes, and they contain lysozyme that will degrade the bacterial cell wall. Okay, so for the next question B, describe how mucous membrane protects the human urinary tract. Okay, the urinary tract. So the lining the urinary tract, it is consists of epithelial cells, and this cell can secrete will secrete substances such as antimicrobial agent. Okay, and then the urine produced is also acidic. So these two factors, okay, secretion from epithelial cells such as antibacterial agent and also acidic urine will inhibit the growth of bacteria, okay, in the urinary tract. And also the urination's action, it provides flushing action, okay, that prevents pathogen from colonizing the urinary tract. Okay, flushing action to flush away, to wash away, okay, the pathogen of the urinary tract. And then the next question, how does the skin works as the first line of defense or the external barrier? Okay, so your skin will have oil, okay, oil and also sweat glands. So this gland will make the skin condition acidic of pH around 3 to 5. Okay, so this acidic condition of the skin will inhibit the growth of pathogen, and at the same time the skin is also home to normal flora. Okay, this will prevent the growth of the bad bacteria or the pathogen, okay, on your skin. Okay, so that's done with question number 5. The total mark is 5 marks. Okay, and then for question number 6, give one example of antimicrobial protein secreted by virus infected cell. Okay, and explain its mechanism to inhibit viral reproduction. So as you can see, this question, it is worth 3 marks. Okay, so for the first point, the most important point that you must include in your answer is the example of the antimicrobial protein, which is in this case is interferon, which prevents, okay, the the viral reproduction. Okay, so this is interferon. If you look at in your notes, okay, the antimicrobial protein, it falls under the second line defense. Okay, second line defense or internal defense. So there are two types of antimicrobial protein actually, which is one is the interferon that inhibit viral reproduction. The other one is a complement system or complement protein. Okay, so for this question, it is interferon. So interferon works. How does the interferon works? Okay, so infected body cells, okay, infected body cell of virus that enters into the host cell will trigger, okay, will trigger the gene that code for the synthesis of interferon to be activated or turned on. Okay, so once this gene is activated, as we have learned during chapter 1, okay, the activated gene will undergo transcription and also translation. It produces mRNA. From mRNA, it produces the protein, which is in this case is interferon. So the interferon produced by infected body cell, okay, infected body cell, will then migrate or find the uninfected, okay, the uninfected cell, the healthy cell. So the interferon will stimulate the healthy cell or the uninfected cell to produce antiviral protein. So again, for the uninfected cell to produce antiviral protein, it will activate gene that code for the antiviral protein to undergo transcription and also translation to produce the antiviral protein. And the function of this antiviral protein is to block viral reproduction in the healthy cell, okay, in the uninfected cell. So this will prevent cell-to-cell spread of the virus. It prevents virus to reproduce in this healthy host cell, uninfected host cell. Okay. Okay, so for the next question, question B, again, please, if before we move on to question B, please make sure that you know how complement okay, system or protein works. Okay, basically it is a group of protein. When it is activated, it will trigger a reaction that will cause the protein to be embedded in the membrane of the virus or pathogen to form pores. Okay, so formation of pores on the membrane of the pathogen causes water or ions enter into the pathogen, causing the pathogen to burst, swell, and then burst. It lyses. Okay. And then okay, for question B, figure 12, okay, shows adaptive immune response. Adaptive immune response is the third line defense. Okay, third line defense. So you can find this diagram in your lecture note. It is actually, if you refer to your lecture notes, it is actually this diagram. Okay, so I some scribble on my notes here. It is actually a simplified version of this diagram from your notes. Okay, so as you can see, you have X. Okay, what is X? The function of X, as you can see here, it is to engulf antigen. So obviously X will be phagocytes, okay, that will engulf the antigen and becomes APC. It has to become APC because the APC molecule, the APC, the antigen presenting cell, will interact with the helper T cell. Okay, so this APC, it can be macrophage or dendritic cell. Okay, dendritic cell or B cell. Okay, so these are phagocytes that when they engulf the pathogen, they become APC by presenting antigen fragment using class 2 MHC. Okay, so APC will present the antigen fragment to helper T cell. So the interaction between APC and helper T cell will cause APC to secrete a signaling molecule, which is cytokines. So the cytokine is used to activate the helper T cell. So the helper T cell can now, when it is activated, it will secrete its own cytokine that has several functions. First, the function of the cytokine released by the helper T cell is to activate itself, so that the helper T cell can proliferate and differentiate into effector and also memory cell, which it undergoes clonal selection. Okay, so another function of the cytokine, the activated cytokines secreted by the helper T cell, is to activate cytotoxic T cell here, and then also the B cell. You can add here, it is actually cytotoxic T cell. Okay, and then okay, so the B cell, the activated B cell will involve in the humoral immune response or the antibody mediated antibody mediated immune response. Okay, for the cytotoxic T cell, it will involve in the cell mediated immune response. Okay, so the activated B cell and also the activated cytotoxic T cell will undergo proliferation and also differentiation, which is clonal selection here. So the many B cells that have been produced through proliferation that has receptor specific for the initial antigen exposed earlier. Okay, so the activated B cell will undergo clonal selection to produce two colonies, which is the effector B cell. The effector B cell is called as plasma cell, and the other one is memory cell. Okay, same as the helper T cell, when it is activated, it undergoes clonal selection to produce effector helper cell or activated helper T cell, and also memory helper T cell. Okay, so the memory cell, it has a long lifespan. It will be stored in the lymphatic system, in the lymph nodes, for many years to protect the body against infection by the same pathogen again in the future. Okay, so the plasma cell will secrete Z. What is Z? Z is actually antibody. Okay, Z is antibody, right? So the antibody will circulate in your body fluid. Okay, your body fluid, it can be in your the virus or the pathogen can be in your blood. Okay, they can be in your lymph, or it can be in the interstitial fluid surrounding your body tissues. Okay, so the activated cytotoxic T cell in the cell-mediated immune response, when it is activated, it undergoes clonal selection to produce memory T cell and also the effector or activated, okay, effector or activated cytotoxic T cell. Okay, so the memory cell again will be stored in the lymph nodes for many years to protect yourself against infection by the same virus. Okay, so the effector cells, okay, effector cells. Okay, so the activated T cell will work at the time of the infection. So all the effector cells, the effector B cell, the plasma cell, the effector cytotoxic T cell or the helper T cell will work at the time of infection. Okay, so basically for the cell-mediated immune response, it will protect the body against intracellular pathogen. Okay, intracellular pathogen, which is pathogen that infected body cells. Okay, so the cytotoxic T cell will basically destroys the infected body cells. Okay, exposing the pathogen, deprive the pathogen of a place to reproduce. Okay, and then for the humoral immune response, it will protect the body against extracellular pathogen. Okay, extracellular pathogen, that which is pathogen that circulates in your body fluid, your blood, lymph, interstitial fluid. So when the extracellular pathogen encounters antibody, so antibodies will bind to the pathogen and causes reactions such as neutralization, opsonization. Opsonization can be divided into precipitation or agglutination, and also the other one is activation of complement system. Okay, so please refer back to your notes to find on how antibody actually destroys pathogen. Okay, so antibody, you have to remember, it does not directly kills the pathogen, but it will undergo all the reaction that I've mentioned before: neutralization, opsonization, and also activation of complement system to destroy the pathogen. Okay. Okay, so for question number 1, Give one example of cell X. Okay, so cell X are cells that will become APC. So your answer can be macrophage, dendritic cells, B cells. Okay, so any of these three cells will be accepted. Okay, and then state the maturation site of cell Y. So cell Y is effector T cell. Okay, effector or cytotoxic T cell. So cytotoxic T cell is a type of T lymphocyte. So it matures in the thymus. Okay. Anyhow, all these blood cells are produced in the bone marrow. But for T cell, it matures in the thymus. For B cell, it maintains in the bone marrow to mature. Okay. And then question number 3, describe the opsonization action by Z. So Z, it is antibody. Okay, molecule Z is antibody. So antibody will not directly kill the pathogen, but it will undergo or involve in in several reactions that will destroy the pathogen. Okay, this question, it is worth 2 marks. Okay, so describe opsonization is one of the way of how the antibody will destroys the pathogen. Okay, so antibodies will bind to the antigen of a pathogen. Okay, antigen of a pathogen, and then once the antibody binds to the antigen of a pathogen, this causes the antigen to be readily presented to for phagocytes to come and then engulf. Okay, to come and then engulf. So present a readily recognized antigen for phagocytes. Okay, or it involves in the process of precipitation or agglutination. Opsonization, okay, it has it involves in two ways, which is one is precipitation and the other one is agglutination. So for example, you have bacteria or virus circulating in your body fluid. If the and then antibodies will bind to the pathogen and causes the pathogen to clump together. Okay, so that is agglutination. Okay, agglutination. So once the pathogen are clumped together, then phagocyte can come and then triggers phagocytosis. It triggers phagocytosis. Same goes to precipitation. Okay, so but for precipitation, it involves the antibody to bind to dissolved antigen. Okay, dissolved antigen causes the dissolved antigen to precipitate. So the precipitation of the antigen, of the dissolved antigen, also triggers the process of phagocytosis by phagocytes. Okay, so that is one of the way of how antibody will destroy the pathogen. Okay, antibody will not directly kill the pathogen, but it undergoes this kind of mechanism. Okay, the other way of how antibody destroys pathogen is through neutralization. Okay, and the other one is through activation of complement protein. Okay, activation of complement protein. The interaction between antibody with the antigen of a pathogen causes the complement protein to be activated, and the activated complement protein will trigger a cascade of reaction where the complement protein will be embedded in the membrane of the pathogen, causing the formation of pores. Okay, so maksudnya nanti sel pathogen tu akan berlubang-lubang with the formation of pores from the complement protein, and causes water and also ions to enter into the pathogen cells, and causes pathogen cell to burst, to swell and then burst, lyse. Okay. Okay, so for question number 3, here, state the effects of second exposure to the same antigen on the concentration of Z and gives its importance. Okay, so Z, as you know, it is antibody. So the question wants you to explain for 2 marks. Okay, what happened? Okay, what happened to the concentration of the antibody during the second exposure? okay, to the same antigen. So when you are infected by a certain pathogen, okay, for the first time, the response is slow. So that is primary immune response, because your body needs to build up your immune system against that pathogen. So your body needs to activate certain B cell, certain T cell, so that the activated B cell, T cell can undergo clonal selection to produce memory and effector cell. Okay, so it takes a long time. So during second exposure, your body has already produced memory cell. Okay, memory B cell, memory T helper cell, memory T cytotoxic cell. So your body has already produced the memory cell against that specific pathogen. Okay, the same pathogen carrying the same antigen. Okay, so because your body has already produced the memory, let's say B cell, okay, B cell. So the memory B cell can straight away be activated to produce the effector cell. Okay, the effector cell for B cell is the plasma cell, and the plasma cell can straight away produce antibody. Okay, so therefore the concentration of the antibody increase. So you have to write this in full sentence. Okay, so the concentration of the antibody increases. So therefore for secondary exposure, okay, so the response is obviously faster, a lot stronger or rapid. Okay, or it takes a short time, which is about 2 to 7 days. Okay, compared to the first exposure, primary immune response, it takes a longer time. It is slower. Okay. Alright, so for question C, figure 13 shows an allergy reaction that occurs in human. Okay, it occurs in human, allergic reaction. Okay, so as you can see, the reaction that involves during allergic reaction, it involves basically molecule G. Molecule G are antibodies of type IgE. Okay, so you have several different types of antibodies actually, but the one that involves in allergic reaction is IgE. Okay, so the IgE molecule, this antibody is produced by specific B cell. Okay, so B cell, when your body first exposed to the allergen, okay, the possible substance from outside that enters into your body. Allergens can be certain foods, or dust mites, or pollen grain, or fungi, okay, whatever that can cause allergic reaction to a person, even cosmetics. Okay, so during first initial exposure to the allergen, your body does not produce the allergic reaction. Okay, why? Because during this time, during the first initial exposure to the allergen will bind to the receptor of the B cell. Okay, the binding of the allergen to the receptor of B cell causes the B cell to be activated, and forms or produce plasma cell. Okay, mekanism dia sama juga yang macam kita tengok mekanism yang ke atas ni kan. So initial exposure engulfed by APC, and then activated by helper T cell, B cell is also activated. Okay, so the mechanism is the same. So the plasma cell produced will secrete antibody for the antigen on the allergen. Okay, so when you are exposed again to the same allergen, okay, this time when the allergen enters into your body, the IgE molecule, okay, the antibody molecule that was released earlier, it binds to this cell, mast cell. Okay, kita rewind dulu. Okay, plasma cell secretes the antibody. Antibody then goes to mast cell. Mast cell is host cell punya. Okay, so mast cell will have receptor. Okay, receptor where the IgE will bind to. Okay, so in the mast cell, it has many vesicles containing histamine. It has many vesicles containing histamine. So, rujuk balik awak punya nota dalam inflammatory response. Okay, mast cell tu apa dia? Histamine tu apa dia? Okay, it is a signaling molecule that is released, okay, by the mast cell. So, this is during the first exposure to the allergen. Okay, initial exposure to the allergen. But when you are exposed again to the same allergen in the future, okay, so the allergen will then bind to the antibody that binds to the mast cell during the first initial exposure. So allergen will bind to the neighboring antibody on the mast cell and causes cross-linking of neighboring antibody. So this reaction will cause the mast cell to release histamine, okay, from the vesicle. If I'm not mistaken, the process we call it as degranulation. Okay, degranulation. So vesicles in the mast cell containing histamine will be released. Okay, so the histamine, one of the effects of histamine is that it causes vasodilation of blood vessels and also increases the permeability of blood vessels. So more blood will flow to the affected area. So when, for example, you inhale pollen, okay, so it causes you to sneeze, for example, okay, so it causes that area to be extra sensitive. Allergy reaction is hypersensitivity towards allergen, okay. So okay, question number 1 here, identify the type of antibody labeled as G. Okay, G is, as I said, IgE, okay, a type of antibody. Okay, so give one common symptom caused by H. H is histamine. So histamine, it causes more blood to flow at the affected area and causes formation of rashes, runny nose, okay, mucosal edema, sneezing, mucus secretion, watery eyes, and itchy eyes. Okay, so those are the symptoms of a person that has allergic reaction. Okay, question number 3, the last question here. Based on figure 13, describe the mechanism of an allergic reaction. Okay, describe the mechanism of an allergic reaction. So I have explained all that. Okay, yang tadi tu sama je. So this question, it is worth 3 marks. Okay, so initial exposure to the allergen or antigen of the allergen, this triggers the plasma cell to produce and secrete antibody IgE. So you must remember this one, IgE. So and then IgE bind to the mast cell because mast cell has receptor for the IgE to bind to. And then upon subsequent exposure to the same antigen of the allergen, antigen cross-link to neighboring IgE on the mast cell. Okay, yang ni lah cross-link ni. Okay. So this trigger mast cell to release histamine. Okay, so you have five points there, but the marks is 3. So any of the point mentioned, so you get the full marks. Okay, so that's the end of this discussion for chapter 7. Okay, thank you.